Bismillahirrahmanirrahim. Assalamu alaikum. My name is Dr. Hassan Akbar and I am a clinical physiotherapist by profession. In this video, we will discuss about the most common question asked by the examiner in the therapeutics Viva. अगर आपने मेरे चैनल को अभी तक लाइक एंड सब्सक्राइब नहीं किया तो kindly like and subscribe my YouTube channel. In this video, we will discuss about the difference between muscle spindle and Golgi tendon organ. Muscle spindles are small encapsulated sensory receptors जो कि lie करते हैं within the muscle belly and they are responsive to the sustained quick tonic stretch. And the tonic reflex mechanism was first described by Charles Sherrington and it is an involuntary mechanism leading to the changes in muscle activation and force with its slow stretch. According to a rough idea, approximately 50,000 muscle spindles exist in the entire human body and they are most commonly absent in the facial muscles. As we have discussed earlier, muscle spindles, they are responsive to the stretch. That means change in the length of a muscle. So, the muscle spindles, they receive and convey information regarding the changes in the length of a muscle and the velocity with which the length of a muscle changes to the central nervous system. This is how our central nervous system controls our position and movement of our extremities in space. This whole mechanism is very important for the motor control, maintenance of our posture and stability of our gait. If we talk about the composition of muscle spindles, that include efferent sensory fibers, efferent motor fibers and specialized muscle fibers that are known as intrafusal muscle fibers. Intrafusal muscle fibers has polar ends that are contractile component and the central epitorial portion that is non-contractile component. Intrafusal muscle fibers are innervated by gamma motor neurons that are also known as fusy motor neurons, and to a lesser extent, intrafusal muscle fibers are innervated by beta motor neurons. If we talk about Golgi tendon organ, Golgi tendon organ was a discovery of Camillo Golgi. It is also known as Golgi organ, tendon organ, musculotendinous organ or a musculotendinous spindle. Musculotendinous spindle, it is located at interface between the myotendinous junction of the extrafusal muscle fibers. Extrafusal muscle fibers are innervated by alpha motor neurons that are of a large diameter as compared to the gamma motor neuron that are of a small diameter. They sense the change in the tension placed on a muscle. And we discuss it with the case like if a person lifts an object, it means it places tension on the muscle. The Golgi tendon organ that become activated and they inhibit the alpha motor neuron that causes the innervation of the extrafusal muscle fibers. Up to 1967, it was thought that Golgi tendon organ, they are only responsive to the high tension placed on a muscle. But after 1967, it was perceived that Golgi tendon organ, they are responsive to a very low threshold or to a very low tension placed on a muscle and that can also prevent from the injury. Thank you.